I will be quiet, and I would like to give the word to Dr. Henrik Johansson, our senior scientist and co-developer of the GARD platform. Henrik, the floor is yours.
Thank you, Peter. Always a pleasure. I'd also like to thank you for participating and greet you to this webinar. My name is Henrik. I'm a researcher here at SenzaGen. I have been working with genomics applications and in vitro assay development for almost 10 years now, both in the academia at Lund University and now as a senior scientist at SenzaGen. My goal with this presentation is to create a good understanding of the concepts of the GARD platform, its strengths and limitations, and how it can be applied to create assays for various biological endpoints. And throughout the presentation, the leading example for this will be our lead product, which is GARD Skin for assessment of chemical skin sensitizers. So at SenzaGen, we develop in vitro assays for immunotoxicological endpoints, primarily by using genomics technology. We were founded in 2010 as a spin-out from Lund University, where I did my PhD work at the time. We are based in Lund in Sweden, and we currently employ 20 people. And our lead product is the GARD platform. GARD stands for Genomic Allergen Rapid Detection, and it's a state-of-the-art technology platform for assessment of chemical sensitizers. So the challenge is to design assays for a number of endpoints, and ideally we want them to provide relevant results with a high predictive accuracy, and we want them to be simple to perform. And for various reasons today, it is ideal if they do not rely on animal experiments. And traditionally, there has existed at least a conceptual trade-off between the simplicity of a model and the informational content it generates on which we can base our decisions. So our goal has been to eliminate this trade-off and provide an assay that is both simple and based on in vitro systems and provides sufficient information for the end user to trust the results. Of course, a fundamental understanding of the underlying mechanisms of the biological endpoint studied is a requirement in order to do so. And in this specific case, that involves the intricate response to foreign substances that lead to sensitization. And these biological mechanisms are today quite well understood and are often summarized in what is now known as the adverse outcome pathway of skin sensitization. And upon exposure, skin sensitizers penetrate the skin in order to reach the dermis, where cells of the immune system reside, primarily dendritic cells, or Langerhans cells, as they are also called. They react with endogenous proteins to form so-called hapten protein complexes. And these hapten protein complexes are taken up by dendritic cells, which mature and migrate to local lymph nodes, where they interact with other parts of the immune system, primarily T cells. These T cells will become primed to recognize the specific sensitizer and forms the basis of an immunological memory in the now sensitized individual. And upon a secondary exposure, these cells, together with various other leukocytes, will rapidly react and elicit an immune response, causing the clinical symptoms associated with allergic contact dermatitis. And there are many parts in this AOP that can and has been exploited for the design of predictive in vitro assays. We will focus on the dendritic cells due to their initiating role as decision makers in the cascade of downstream events. And dendritic cells typically serves as sentinels in the periphery, guarding against non-self molecules, or so-called antigens. And upon encounter of such foreign substances, the dendritic cells go through functional and phenotypical changes, enabling them to initiate and orchestrate an immunologic response. So the question is how we best monitor these changes. And we now know that these cells react in a multitude of different ways upon encounter of an antigen. Firstly, they are equipped with various receptors that recognizes foreign material. And these receptors may in turn signal to and activate various self-defense mechanisms of the host. And you might have cellular stress responses, such as oxidative stress or metabolic stress. And finally, they express receptors that allow them to migrate and communicate with other cells of the immune system. And all of these functions are of course controlled by proteins in the individual, and the proteome is of course in turn controlled by the transcription of DNA. So therefore we believe that Genomics, or more specifically gene expression analysis, is a very efficient way to get a holistic view of what is going on in the cell upon a chemical exposure, as it provides an opportunity to measure not only one or a few biomarkers, but all of these events can be monitored simultaneously. So with this knowledge and assumptions, we early on hypothesized that there should exist genes in dendritic cells that are differentially expressed depending on the stimuli, so that they would react differently to, for example, a sensitizer or a non-sensitizer. And if we were able to find such genes, they could be used as predictive tools. So we set out to test this hypothesis by using a reference panel of chemicals for exposure of a myeloid cell line, acting as an in vitro model of dendritic cells. This panel of chemicals included 20 well-known sensitizers and 20 non-sensitizers. After exposure, we analyzed the expression levels of the entire human genome using microarray technology. And by using data-driven biomarker discovery approaches, we were able to identify a set of 200 genes with unique expression patterns induced by sensitizers and non-sensitizers. And we refer to these genes as the GARD prediction signature, or GPS in short. And of importance, by looking at the identity of the genes and the canonical pathways with which they are associated, we could confirm that they are indeed immunologically relevant, as they are largely involved in the mechanisms described earlier. And I will come back to the identities and the biological relevance of the GPS later on in this presentation. So we have now designed a predictive assay based on this knowledge that utilizes the predictive power of these genomic biomarkers. And all protocols are described in detail in a comprehensive SOP, but conceptually this SOP can be summarized in 6 distinct steps, which I will now go through. So first of all, we culture in vitro models of dendritic cells in a cell lab facility. These cells are exposed to any test substance to be assayed. And in the first step, we establish an appropriate input concentration of the test chemical. So we do a dose response viability analysis in order to monitor any cytotoxic effects induced by the test substance. And the goal is to identify the one concentration that induces 90% relative viability of the cells. This allows us to both confirm bioavailability and ensures that cells are not killed too much, which would impair their capacity to function immunologically. Once an appropriate input concentration has been established, we repeat exposures in 3 consecutive and independent experiments, generating biological triplicate samples. And the last biological step is to isolate RNA from these chemically exposed cell cultures. And at this point, the biological cell work is concluded and the phase of analytical work continues. So in the 4th step, we first do a quick quality control of isolated RNA. Then we move forward with every RNA samples in the gene expression analysis, which is performed with the nanostring encounter technology. And this is a step in which raw gene expression signals are generated and the expression levels of each of the 200 genes are quantified simultaneously. So it's worth emphasizing that while all discovery work was done on microarrays or next generation sequencing, the assay has now been transferred to the nanostring platform, which is a very fast and easy to use and resource effective methodology for multiplex gene expression analysis. Finally, once we have the gene expression data at hand, we use machine learning techniques to classify each sample as a sensitizer or a non-sensitizer. And while these data analysis steps are state-of-the-art, no data science expertise is required from the operator or user of the assay. Every licensed lab that has set up GARD in-house will have access to our cloud-based data analysis application. So raw data is simply uploaded into the app, which is cloud-based, and classification results will be generated automatically. For any lab that is interested in setting up the GARD in-house, it might also be good to know that different options exist. Depending on the equipment you may have available, you may choose to transfer all of these steps to your laboratory, or you can just also transfer the cellular protocols and send your RNA samples to us or any other CRO with access to nanostring equipment, which would then send you back the raw data files, and you would still have access to the data analysis app and would simply just upload all the data to generate the results required to finalize the report. So by now we have generated an in-house database of about 150 chemicals that have been assayed using these protocols. If we look at published data, we can conclude that GARD is consistently around 85 to 90% accurate. And according to recent literature, this is the highest reported predictive performance available from both in vivo, in vitro, and in silico counterparts. Of course, these types of figures might vary a bit depending on the subset of chemicals included in a specific study. So in a meta-study by Professor Dave Roberts at the Liverpool John Moores University, a coherent subset of chemicals was explored for which data exists both from GARD and the 3 validated in vitro assays. And in this set of 69 overlapping compounds, Professor Roberts concludes that GARD not only outperforms currently validated methods on a one-on-one basis with regards to predictive accuracy, but it also performs better than a 2 out of 3 integrated testing strategy based on the validated methods. So it is not entirely unmotivated to ask why and how is GARD performing so well. And the honest answer is that we don't really have any complete straight answer, but we can identify certain aspects of GARD that we do differently compared to existing methods. There might be some repetition in these next slides, but I'd just like to emphasize 3 unique technology features of GARD that I think all contribute to the high observed predictive performance. These are the data-driven assay development, the high informational content generated by the assay, and the state-of-the-art prediction model, which is based on machine learning technology. So first of all, the genomic biomarkers that GARD utilizes as predictors were identified by a data-driven approach. And I say data-driven here as opposed to hypothesis-driven. That means that instead of basing our design on a priori knowledge, such as an assumption that the gene expression of a certain co-stimulatory molecule will go up, we interrogated the data generated by a reference panel to learn of what really happened. And as it turns out, co-stimulatory molecules are indeed of great relevance, but that's not the only thing that we found. Instead, we found 200 genes that collectively exhibits distinct expressional patterns when comparing the group of sensitizers and non-sensitizers. We can then use this information to build models that generalize well. That means that while not all genes may be affected in the same way by all chemicals, when studied collectively, these genes could make a distinct separation between sensitizers and non-sensitizers possible. The second unique feature is really a direct consequence of the first, and that is the high informational content of the assay. As we have already established, even when studying only one cellular system or key event of the AOP, the cells do not respond in only one way. That is the same for all chemical exposures. Instead, a lot of different things are going on. So the problem with choosing a too simple model, often relying on only 1 or 2 biomarkers, is that the reality is so complex. And using only very simplistic models seldom captures this complexity, making the information generated not useful in the end. But by adding parameters to your readout, you start building data that better captures this complexity. And in the end, of course, you'd like to see the big picture. And in the case of GARD and the GARD prediction signature, I believe that this is truly the case. In fact, we can show that the genes involved are indeed associated with all of these main functional groups of effects induced by sensitizers and dendritic cells. We capture, for example, the recognition of foreign substances by inclusion of TLRs and the aryl hydrocarbon receptor. We monitor the innate immunity in the form of CD86 expression, among others. And we do also monitor cellular stress responses, as many of our genes are involved in the NRF2 pathway, and so forth. So of great importance here is that even though the genes were identified using a data-driven approach, we can confirm that they are indeed involved in the expected cellular mechanisms related to chemical sensitization, and therefore are of biological relevance. The last unique feature of GARD that I would like to emphasize is the way that we analyze this high-dimensional data, which is based on machine learning technology. And these are concepts that are very well established in many fields of technology by now, and we are in no way the first to do this. And all algorithms that we use are well established in literature, and they are based on open source code. But the general goal of these algorithms is to have a machine learn to recognize patterns from a set of examples, often referred to as a training data set. In our case, the training data set is the data generated by the reference panel of chemicals during assay development. So each sample will have a unique expression pattern over the 200 genes that we measure, and each sample will also be associated with a group belonging as a sensitizer or non-sensitizer. So what the machine does is collect all of the data and identifies patterns that distinguishes the groups. And the model is now considered trained and is ready to be applied to external data, often referred to as test sets. So when the machine or algorithm is then challenged with an unknown test substance, it will classify it as a sensitizer or a non-sensitizer based on the expression profile of that distinct sample, a process referred to as pattern recognition. So the specific algorithm that we use is called a support vector machine. Very briefly, it creates a hyperplane in the 200-dimensional space of the measured biomarkers. And a test substance is then classified by the sign, which is either positive or negative, relating to the positioning in this space. So even though it may sound very abstract, the output from the algorithm is really quite intuitive and easy to interpret. It comes in the form of decision values. So on the right here, you can see actual data from an in-house study. So any sample induced a mean decision value that is greater than 0 is classified as a sensitizer. And correspondingly, of course, if the decision value is smaller than 0, it is a non-sensitizer. And we know that these technologies might sound a little bit scary. They are based on advanced mathematics and software programming. So I just want to emphasize again that no expertise is required by neither the user of GARD or the sponsor of a study to interpret the results. We will provide access to our easy-to-use data analysis application software that is reached through any web browser. And as for the end result, it will be both easy to interpret and highly accurate. So I keep repeating that even though it is state of the art and it is quite easy to actually do, and to prove this, and in order to demonstrate that the assay really is transferable, reproducible, and accurate, we just recently performed an inter-laboratory ring trial with the goal, of course, of reaching formal validation and regulatory acceptance. So we have transferred the assay to 2 naive laboratories, which are Burleson Research Technologies in the US and Eurofins in Germany, and established every part of the GARD SOP in these laboratories. In a first phase of blind validation, we had these 3 laboratories, ourselves included, assess 28 blinded test chemicals. And the results look like this. So these are the decision values for each of the 28 compounds we ran. The details of this will of course be made public. I don't expect you to read everything on the axis, but to summarize, we can conclude that the within laboratory reproducibility ranges from 82 to 89%, with a between laboratory concordance of 92%. And the predictive performance is quite impressive. Also in this blind data set, we have a predictive accuracy of 94%, averaged across the 3 laboratories. So The GARD skin assay, as I've been talking about up to this point, is a binary assay, which means that it classifies a test chemical as sensitizer or non-sensitizer. We do also assess sensitizing potency, and we do so by a complementary biomarker signature, which subcategorizes any sensitizers into 1A or 1B, as defined by the global HART system. So we propose a tiered approach for any test chemical to be assayed, where GARD skin initially classifies a test chemical as sensitizer or non-sensitizer. If it is classified as a sensitizer, you will have the option to subcategorize it into 1A or 1B using the GARD potency prediction model. The GARD potency prediction model will also be validated with the same set of 28 compounds, and in order to ensure statistical power, we have actually added another set of 30 compounds for the lead laboratory to assess. The GARD skin data has been submitted to ECVAM and OECD scientific advisory committees. It is currently being reviewed, and the GARD potency data is being submitted this very month. So we expect the scientific review of this data to progress throughout the year, and if everything goes well, we expect to have an approved OECD test guideline by spring 2019. So, wrapping up the validation status of GARD skin and GARD potency, for the remainder of this presentation, I would like to give a brief update on the SenzaGen R&D pipeline. So far, I have been covering GARD skin and GARD potency assays, but as I have been saying in the beginning of the presentation, we consider GARD to be a versatile technology platform on which you can base a number of different applications. And with applications here, I typically refer to distinct assays for distinct biological endpoints. And the main applications that are currently in the pipeline are GARD skin, GARD air, and GARD protein. I'd like to start by presenting some development work that is going on in the relation to GARD skin, which is mainly about broadening the applicability domain of the assay. So the next really big area that we consider that GARD skin could really have an impact in is medical device. As some of you may know, there is an ISO guideline detailing of how testing of such products are envisioned, and it is done by an extraction process with polar and non-polar solvents. These solvents are then used as the testing material for the relevant assay. We have successfully been working with several projects testing different silicone-based materials, and also an absorption column for blood purification purposes. And we do provide this service already today and are happy to discuss such projects on a case-by-case basis. In the meantime, we are generating data from in-house proof of concept studies aiming to demonstrate GARD skin's applicability in relation to the ISO using spiked materials. And we expect this data to be finalized later this year. In a very related field of applications, we are also exploring the possibility to use GARD skin for testing of complex mixtures. And basically, this is done in a very similar way as described for the medical device ISO, but depending on the nature of the sample, we sometimes need to be a little more creative. In this regard, we are constantly expanding the arsenal of solvents that are compatible with GARD, and successfully assayed mixtures to date include polymerized gels, oils, and UVCBs from oil industry. And this is also something that we are providing today as a service, and are happy to discuss any related projects on a case-by-case basis. So in order to show you an example of what explorations in this area might look like, I'd like to show you some real data on a sponsored project we completed recently. And here we tested a UVCB with very poor water solubility properties. When using our standard vehicles, which is either water or DMSO, you can see that we could not really demonstrate any bioavailability to the compound in well, as we failed to induce any expected cytotoxicity. And as a consequence, we also failed to induce a positive GARD signal, as shown by the GARD decision value. in the bottom plots. So we explored different options on a trial and error basis until we could demonstrate bioavailability when using a mixture of DMF and glycerol as a vehicle. And here we were also able to demonstrate that the compound was indeed a sensitizer, which also correlated well with experience from in vivo data. The last thing that I just want to mention relating to this field is that while we do consider GARD to have a very broad applicability domain, we are constantly working to both improve ourselves and monitor our weaknesses. And to this end, we are collaborating with researchers at LASA Limited to identify data gaps so that we can subsequently fill those gaps to demonstrate the usefulness of an even larger chemical space. Finally, in an experimental setup, we are also exploring the ability to integrate a framework for conformal prediction into our classification algorithms. And if this is successful, this sort of framework would provide a warning if a test substance is outside of the applicability domain based on the similarity of the test substance to the training data set defining the prediction model. So I'd like to move on now to the next GARD application, which is GARD Air. This is an assay in development that aims to predict the ability of chemicals to induce both respiratory sensitization and irritation. Currently, there are no validated or even widely used assay available for assessment of chemical respiratory sensitizers. And this is at least in part due to the relatively large knowledge gaps relating to the molecular mechanisms involved with respiratory sensitization compared to the case of skin sensitizers. So this means that a complete adverse outcome pathway is currently not available. But what we do know in terms of an AOP is that different subtypes of T cells are likely involved in respiratory and skin sensitization, and that the preferred mechanism of action is likely involving T cell populations differentiated into either Th1 or Th2 phenotypes. We also know that activation of specific T cells is dependent on antigen presentation, most likely by the dendritic cells. So one very relevant question is, do antigen-presenting dendritic cells transmit different signals to T cells depending on the type of hapten protein complex presented? And if that is so, can we detect these differences in the induced transcriptional profiles of stimulated dendritic cells? So again, in order to investigate this hypothesis, we utilized an experimental setup equivalent to that of GARD skin development, which means that we used a reference panel of known respiratory sensitizers for stimulation. And again, using a data-driven analysis approach, we were able to identify a biomarker signature that is able to separate respiratory sensitizers from controls. And this further demonstrates the advantages that data-driven analysis provides, as useful results can actually be achieved even though the molecular basis of the endpoint is not entirely known. And the validity of these biomarkers has been confirmed using an external test set where we were able to predict 5 respiratory sensitizers from 20 non-respiratory sensitizers with an accuracy of 84%. And it's of specific interest here that the group of non-respiratory sensitizers actually include skin sensitizers, which are here accurately predicted as non-respiratory sensitizers. And this shows that GARD Air is indeed specific for respiratory sensitization. This project has gained a lot of positive attention, and we were fortunate enough to be granted funding from the EU Framework Horizon 2020 to explore the validity of this project further. And the project aims to take these findings all the way from the R&D stages that we're currently in to a finalized product, best case scenario, including a method transfer and validation. And we also incorporate the hypothesis that the GARD platform could be utilized to identify and discriminate also respiratory irritants, so that a conceptual product would provide assessments of double biological endpoints from the same sample. And the underlying R&D work behind this hypothesis is work performed in collaboration with AstraZeneca, who is particularly interested in this endpoint within their drug discovery pipeline. And the training data set used was actually provided by AstraZeneca and consists of active pharmaceutical candidates from their pipeline with very complete annotations and previous information from both in vivo studies and clinical trials. And based on this reference panel, we were able to identify expressional patterns relating to the irritating effects observed in human. We have already had an opportunity to apply both of these models on external test data provided by a sponsor. I can unfortunately not declare the source or substance identities of the test substance at this point, but we can conclude that GARD air remains a very promising assay for respiratory sensitization, as we classify 6 out of the 7 positive controls correctly, as well as the negative controls, including skin sensitizers. And similarly, the data generated by the irritancy model correlated very well to the outcome expected by the sponsor. And the collaboration with the sponsor will likely continue, and we expect that this data will be published and fully disclosed within this year. And with that, I'd like to move on to the last application of the GARD platform, which is GARD protein. As the sound may suggest, this is an R&D project towards an application of GARD for assessment of protein allergenicity. In a first study, we investigated the induced transcriptional profiles induced in vitro dendritic cells in response to 8 industrial proteins with known allergenic effects. And again, using a data-driven approach, we identified a biomarker prediction signature specific for allergenic proteins. And in this principal component analysis, we show how proteins cluster separately from negative controls. Following this pilot project, we did not really have any opportunities to test its functionality until we explored its applicability on food allergens as commissioned by a sponsor. So in a follow-up study, we investigated 2 homologous versions of tropomyosin from different species. One from shrimp, which is known to have allergenic properties, and a non-allergenic counterpart, which was isolated from pork. And we used GARD protein as defined to classify these samples. And it turns out that the shrimp tropomyosin was accurately classified as an allergen, while the pork tropomyosin was classified as being negative. So these results serves as a first indication that GARD protein is functional. And while it is still in the R&D phase, we are exploring opportunities to take this research further together with any interested partner. So I'd like to conclude this presentation and summarize a few take-home messages. We believe that genomics technology allow for convenient and accurate safety assessment of chemical sensitizers. And it does so by facilitating simple models that effectively monitors a complex reality with a high information content that allows you to see the bigger picture. And this is, we believe, what provides a robust assay which is consistently proven functional with a high predictive performance. The platform is versatile, which allows for a broad applicability domain, as exemplified by the different applications I have exemplified here today. And my last take-home message is that this technology is readily available today. And if you are interested to use it either as a sponsored project, or if you would like to collaborate with us on any of the scientific and experimental assay we have in our R&D pipeline, please don't hesitate to contact us. And you will have all the details of contact either through Peter or our website. So I'd like to move over to Peter to conclude this session and maybe answer some questions that you might have.